This is lab number 8, enzymes. So enzymes are a group of proteins that are going to help reactions occur. Okay, it's going to speed them up. Okay, reactions are a group. Enzymes are a group of proteins. Okay, and proteins have a lot of different functions. In lab 4, Professor Russ, and also in our lecture, we were talking about proteins, and they are organic molecules. They're one of the important ones. And we were talking about how the shape of them kind of helps determine what their function is. Okay? So enzymes have a specific shape because of the order of their monomer, which is amino acid. Okay? And they're going to help fold and bend and then create a certain kind of area where something can stick to it, to bind to it, okay? So we're gonna get to all that in just a second. But that's where we're kind of going, and we're gonna look at things that affect how an enzyme works, okay? As we go through the lab. So enzymes are proteins, and they are called biological catalysts, okay? So in the few sentences that I said just a second ago, I said they help speed up a reaction. So a catalyst is some kind of molecule or something that speeds up a reaction. Okay, makes it occur faster or easier, kind of like working smarter, not harder type of scenario. Okay, and it's going to do that because it's going to bind to a substrate. Okay, the substrate is just what it's going to, what's going into the reaction. Okay, so we have this diagram here at the beginning of our lab. Okay, and it's kind of simulating how an enzyme would work. So the gray structure here is the enzyme. And it has a specific little shape right here, okay? That shape is called the active site, okay? So I said the amino acids determine the type of protein that it is, and it also will determine how it folds and bends and creates a shape, okay? So you're gonna give the protein or enzyme as a beaded necklace, okay? And so you have each bead as an amino acid. If you lay that necklace down on the counter, it might kind of wave around a little bit, right? And that's going to be kind of like a little bit of attraction, okay? But if you just kind of clump it all together, and now you have this ball of a necklace, okay? Somewhere in there might be an area for something to bind to, okay? So we're kind of thinking of a 3D structure of this molecule. So this is where it's folded in because of hydrogen bonding, okay? Small, slightly attracted hydrogens to each other, making that fold, and now we have this active site. Okay? So here we have this. This is where it's going to be a lock and key for whatever it's going to help for that reaction. Okay? So the substrate and the reactant, what goes into a reaction, are going to be the same thing. Okay? So substrate or reactant, we're going to see that word in just a second. It's going to bind to that active site and form a enzyme substrate complex. The enzyme and the substrate together, okay? It's going to facilitate it into this reaction, guide it through, okay, like a guided tour, into this reaction to help it occur, right? So we have green substrate here, and now this reaction occurs. And now we have this blue-red, okay? And then a blue and a red. At the end of the reaction, we get a product. What did we produce from that reaction? We produced this blue molecule and this red molecule from these that were stuck together as a green molecule, okay, just for our example. At the end, this enzyme is exactly the same as it was right here. So the enzyme doesn't get used up, okay? It doesn't change, it doesn't break down. It helps guide it into the reaction, makes those products, and then it can recycle and be used again. So I said just a second ago that the enzyme is going to have a 3-dimensional shape, that necklace folded up or knotted up analogy that I talked about. Okay, enzymes are really specific, and they are going to work for very specific reactions. They're going to bind to specific substrates. Okay. So it's kind of a lock and key is what we call it. So if I take my car key and I put it into my car ignition, it should start my car. But if I take my car key and I try to start your car, hopefully, or it should not start your car, okay? It's not designed for your car. It's not structured for your car ignition, okay? So it's very specific, okay? I also said that they are catalysts. That was the very first sentence of the lab. A catalyst is a compound or a substance that's going to speed up or facilitate a reaction. Okay? So here in the next part we have a graph. And in this graph it shows the activity, okay, with and without an enzyme. So here is our reaction. Okay? If we did not have an enzyme present, it's going to need this much energy for that reaction to occur. You have to input that much energy. Okay? But our systems, or organism systems, want to work smarter, not harder. Okay? They want to maintain homeostasis. They don't want their core temperature or their body temperature to change very much. Okay? And so they don't like to waste energy if they don't have to. Okay? They just want to work more efficient. And so with an enzyme present, we're only going to need this much energy inputted to get that reaction started. Okay? That input energy is called activation energy. So here's this waiting to happen, and here's the spark plug to start your car, to start the reaction. You need this much energy for that reaction to occur, okay, without an enzyme. But if we have an enzyme, we only need this much. Okay? So we go from here with the enzyme, okay, to here, we only need this much. Okay? Versus this much. And on a graph like this, it doesn't look like a huge deal, but in your body, every single molecule, energy molecule, matters because you have reactions happening all the time, everywhere, and so do other living things. And so you want to be smart about how you are expending that energy. Okay? So we have enzymes. Okay? They're going to help make that reaction occur smarter, right? If you had to move a whole apartment full of furniture, are you going to move it by yourself? Right? Or are you going to enlist a friend or two, right? A friend, an enzyme, to help that go a little bit faster, a little bit easier. Okay? So same type of thing. Okay? So the activation energy is lowered when an enzyme is present. Okay? It lowers the activation energy required for a reaction to occur. Okay? It lowers it. Okay. So our lab today is going to look at an enzyme called catalase. Move that down. An enzyme called catalase. Okay? And catalase is an enzyme. It ends in ASE. Okay? That's a good indication that it is an enzyme. We're going to see some enzymes later when we talk about DNA transcription and replication and things. So ASE is usually an ending that refers to an enzyme. Okay? So it is an enzyme that's going to react with or help the reaction, the breakdown of hydrogen peroxide, H2O2, hydrogen peroxide. Okay, hydrogen peroxide, we'll see that in a second. And it's going to help it break down into water and oxygen molecule, which is going to be released as oxygen gas. Okay, so hydrogen peroxide is stored in a brown bottle. You can buy it in a brown bottle. Okay, why is it in a brown bottle? Does anybody know? It's in a brown bottle because hydrogen peroxide will slowly break down and light will help that occur. Okay, and so it's stored in a brown bottle in the dark, okay, to slow down the natural breakdown of hydrogen peroxide. Okay, if you open that up and you stared at that, it would just look like a clear liquid. Okay, it's if you pour it on a cut, some people do that, some people are sensitive to it. So if you pour it on a cut, it's the one that will bubble. Okay, that's hydrogen peroxide. So it'll naturally break down, but if we add this enzyme, okay, catalase, then we're going to speed that reaction up. We're going to facilitate it to go faster. Okay, and so we're going to demonstrate that. So we're going to look at this reaction, the breakdown of hydrogen peroxide into these two products. Okay, so the first question is, I just gave you the answer. What are the products of this reaction? The products are what you produce. What is produced is after the arrow. These are the products. Okay, so the products are what you produce at the end of the reaction. So you have H2O, which is water, and you have O2, which is oxygen, and it's usually in a reaction like this released as gas. Okay? What is the enzyme in this reaction? I also gave you that a while ago, right? You should have said just now, catalase. Catalase is the enzyme. ASE refers to a substance or a particle, a molecule that is an enzyme. Okay? What is the substrate for this reaction? Well, we already labeled these, this, and this, and this. So by process of elimination, you could easily say, this is all I have left. Okay? H2O2, which is hydrogen peroxide. Okay? If this was first, right, what substrate? The substrate is going to bind to the enzyme to facilitate and speed up, lower the activation energy of this reaction to break down that substrate. Substrate and reactants are the same thing. Okay? We can also say this is also called reactants. Okay? Substrate and reactants are the same thing. They're interchangeable. Okay? What reacts in the reaction? And what do we produce? Okay? Substrates. Substrate is usually the term used for the reactants when it is involved with the enzyme. Okay? So they're reactants, but they're called substrates, a little more specific when we talk about an enzyme. Okay? What will you observe in this reaction as it proceeds? We're looking at hydrogen peroxide, a clear liquid, with an enzyme, so I'm going to hold up the beaker, that's also a clear liquid. Okay, the enzyme is dissolved in that water. And we're going to be looking at water, which is also a clear liquid. And then we have this oxygen gas. So that gas is going to release in that liquid, and it's going to look like bubbles. A lot of my students in previous semesters said it looked like carbonation, right? Like Sprite, right? Bubbling up. And we'll see that in just a second when we do the lab. Okay? So we're going to be looking for bubbles. That means that we are releasing this oxygen gas. Okay? That means that it is going faster. If I just open this bottle and look in, I'm not seeing that. Okay? But if you were using it as a disinfectant to clean, okay, something, you're going to see those white bubbles. Okay? That's the oxygen gas releasing. Okay? All right. So let's move on down. I'll erase that. The first procedure is 8.1, and we're going to look at this reaction, and we're going to test to make sure our enzyme works. Okay? In one of the previous labs, Professor Russ talked about positive controls and negative controls. Okay? So we're going to look and see if all of our reactants, our substrates, and our enzyme are working, okay? And to see what this reaction looks like, okay? So we're going to have 3 test tubes, okay? In the first one, we're going to have catalase and hydrogen peroxide, okay? In the next one, we're going to have water and hydrogen peroxide, and then catalase, the enzyme, right? And then an alcohol, ethanol. So the first one, we're going to have the enzyme and the hydrogen peroxide, which is, I know it's going back, exactly that reaction. The enzyme, catalase, and hydrogen peroxide. Okay? So I'm going to do that one here in front of you. Okay? Test tube 1. So I have the catalase in here already. Took my 20 drops of my enzyme, and I put it into this test tube, okay? And then I have pre-measured 5 milliliters of hydrogen peroxide, okay? And I'm going to see if I can maybe in front of my black jacket, might be helpful, right? I'm going to put those together and hopefully we see that reaction, okay? So I add that hydrogen peroxide, and right away we're seeing bubbles. You able to see that on my? Let's try that one. Let's try that one. All right, so we can see it's fizzy, right? There are a lot of bubbles occurring, okay? Those bubbles are oxygen gas being released, okay? The enzyme catalase is attaching to and speeding up, attaching to hydrogen peroxide and speeding up the reaction of it breaking down, okay? And it breaks down into water, which is clear liquid, right? And releases oxygen gas, and that's all the bubbles, okay? All right, so that's the reaction that is occurring, okay? So the key says for us to say if there's a high number of bubbles, for us to put 3 plus signs. So there are a high number, a high amount of bubbling, okay? That reaction is occurring very rapidly, okay? What does that mean? That means that that enzyme is working and speeding up the breakdown, okay, of hydrogen peroxide. Okay. Is speeding up the breakdown. Oops. Of hydrogen peroxide. Okay, the enzyme is working. The enzyme is working. Okay, so it's kind of like a positive test because we were trying to test the enzyme catalase and see if it actually worked on hydrogen peroxide. But it also is our experiment because that's the equation that we were seeing, the reaction that we were seeing earlier at the top. Okay. If we put hydrogen peroxide and water together, and we did this hydrogen peroxide and water together, we're not seeing any bubbles. It just looks like a clear liquid, okay? Compared to the one that's bubbling. Yeah? Okay. And so we're seeing that there's no bubbles, okay? It's still probably slowly breaking down because I said hydrogen peroxide is stored in a brown bottle and it reacts with light and it will slowly break down. So if someone at your house used the hydrogen peroxide and they left it open, right? Or even left the top open, that hydrogen peroxide is going to break down faster. And then if someone needed it to clean something, it's going to be more water, right, than hydrogen peroxide, okay? So you want to make sure that you close that. But if it's just water and hydrogen peroxide, it's just clear, just like I would see if I looked inside of this brown bottle, okay? So no bubbles, okay? And so there's no catalase present, okay? Hydrogen peroxide, not, I want to say not breaking down, but in reality it's so slow that we're not seeing it, okay? So I'm just going to say not breaking down. Visibly. Visible, right? Not visible. Okay? It's not visible that we can see that that's breaking down. Okay? There's no enzyme to speed that up, so we're not seeing those bubbles. Okay? Same thing with alcohol. If you change the pH a little bit, or if you have it in a different, okay, and we just use ethanol out of a little bottle, a sample that we have here, okay? This is, I'm trying to get this folded. And then this is already slowing down a little bit, okay? Right? Where you can see bubbles and no bubbles, okay? We can also see that this is slowing down, okay? We'll talk about that in just a second. So the ethanol, again, we're not having that enzyme speed up that reaction. So again, we're not seeing bubbles. And then hydrogen peroxide not breaking down. Okay? Or that reaction is not sped up. Okay? And you can word this in your chart a lot of different ways. Okay? It doesn't have to be exactly like mine. I can say reaction not fast enough to see, or no enzyme present, so it's not facilitating the reaction. There's a lot of ways to word this. Okay? Bubbles, the reaction is sped up, there is an enzyme. Okay? No bubbles, the reaction is not sped up. Okay? No bubbles, no rapid breakdown of hydrogen peroxide. Okay? All right, so that is 8.1, and this next part is going to lead into, I'll try to get that all in one, okay? Okay, erase this. So this aside. I have the next one here, and we're going to put the hydrogen peroxide in. Okay, we have 3 test tubes. All right, so we have 3 test tubes in this next one. Okay, and we're going to be looking at temperature. Okay, but before we do that, I'm going to talk about enzymes, and because they're specific. I'm gonna wait. Okay, because they're specific, Professor Russ is getting the cold one out of the refrigerator for me, so I don't have to go off screen. Okay, so enzymes are very specific, lock and key, of what they will attach with, okay? Like the key of your car, the key to your door, okay? They also like to work in a very specific range, okay? And so if your core temperature or an organism's core temperature gets too high, okay, it can damage that enzyme because it's a protein, okay? Or if it gets too low, it can slow it down or it can damage it, okay? And so the first one that we're going to look at is temperature, okay? And so if something happens to that structure, okay, it's really easy to break those hydrogen bonds, okay? And the hydrogen bonds are what helps that enzyme fold and make that specific shape, okay? So if I were to change the temperature of where it likes to be, okay, where it's formed and structured to be, I can unfold it, okay? Unfold it completely. And if I do that, it can't go back, okay? So we've all probably seen either on TV or we've done it or we've seen someone else do it, cook an egg. Okay, you take that egg, you crack it open, and you put it in the pan, right? Or even if you just boil an egg, okay? Once you cook that egg, you have changed that protein, right? Albumin, you saw that in lab 4, the protein, you tested it for protein that turned purple, okay? When you cook that, and now it's that nice white part of the egg, okay? You can't take that and put it back into the shell as the this is liquid, okay? You've changed that protein. You've heated it up by cooking it, okay? You've raised the temperature, okay? And you've denatured it, you've changed it, you've cooked it, okay? And so the same thing would happen in organisms if it gets into a different area that has a different pH, okay? So enzymes in our stomach, okay, in our gastric juice, okay, gastric enzymes, are close to like a pH of 1 or 2. Okay, but in our blood, those enzymes are a pH of 7. We'll get to pH, okay, and or we've talked about pH already. And so they'll work in different things, okay, or if your fever goes too high, okay, it's going to change those things. I'm getting ahead of myself with the pH, okay. So here we're going to look at, does temperature affect the activity of an enzyme, okay. So we have 3 test tubes, okay. And the first one we put in the fridge, okay, in the refrigerator at 4 degrees, okay? And then we had one sitting at room temperature, and then we warmed up one in a water bath, okay? And so we're gonna look and see how temperature may affect the activity of that enzyme. Get this out of the way, okay? So I'm going to hold this behind here. See how talented I am, okay? Hold this behind here. This is the 4 degree, this is the cold one, Okay, so we put the enzyme in here, we put it in the refrigerator, and now I'm going to pour my hydrogen peroxide in. And it's going to be little bubbles at first, and then it might getting going. But we do see bubbles. This next one, let's see if I can put them together, is the room temperature. It's the room temperature. Let me pour it in and then I'll raise it back up. This is the room temperature. Okay? This one's warmed up now, and so it looks kind of close, but at first, it didn't have the big bubbles like this one did at the beginning. I'm trying to get them right next to each other. Okay. So at first, the cold one was slow, and then the reaction went. So it's like when it's cold outside, you kind of slowly get out of bed, right? Okay, we slowly get out of the bed. It's like, oh, I don't really want to get going. Okay, fine, I'll go. Okay, that was the cold one. And then the room temperature looks exactly like we did in the first procedure, where it's water and hydrogen peroxide, okay, or catalase and hydrogen peroxide together, okay? All right, the last one, I'll put the first one down. So this is the room temperature, okay? The last one, I have another hydrogen peroxide, I need one more. Huh? Oh yeah, it's already done, sorry. Sorry. The last one we put in the heat, we put them together and then we put it in the heat and we pour the hydrogen peroxide in, okay? And this last one doesn't have any bubbles, no bubbles, okay? Why does it have no bubbles? We heated it up like cooking an egg, and so at the very top paragraph on the screen right now, we denatured it. We denatured it. That heat broke down the hydrogen bonds of that enzyme, unfolded that active site where the hydrogen peroxide would bind to it, okay? It unfolded that, and so now it can't bind to it. So you're just getting water and broken up protein, okay? And so that enzyme doesn't have its shape, its specific shape, to make that reaction go like it did at the room temperature. Okay, like it did at the room temperature. Okay, and so we can see that enzymes are affected by temperature change. Okay, so we saw, I'm going to put a lot because this is the same as what we had in the very first one. This is the room temperature, 40 degrees. Okay, and we saw rapid reaction. Okay, rapid breakdown with hydrogen peroxide, okay, releasing oxygen gas, okay? In the 4 degree, the first test tube, the slow one to start, the one that Professor Russ brought into me, right? We saw slow bubbles and then a lot of bubbles, okay? Maybe I'll just put one, put an arrow, okay? Lots of ways to write that. But we saw slow or small, smaller bubbles, slow reaction, smaller bubbles, and then as it warmed up, that's why I was saying maybe not yet, because it probably got a little warm sitting here while I was talking, okay? Then it had rapid reaction, okay? Lots of bubbles, okay? Lots of bubbles. Okay, and right now, if we looked at those again, they would look almost exactly the same. Okay, sorry, impromptu. Okay, they would look about the same. This is test tube 1, the cold one, and this is the room temperature. So they look about the same right now. Okay. So what does that mean? Oh, and then the 90, the hot one, the last one had no bubbles. No reaction that we can see, visible, and that's because we denatured catalase. Denatured, we cooked it, right? Like cooking an egg, okay? We broke down the structure of this enzyme by heating it up. So the next part says draw a graph. Okay, graph your results. Okay, if we were saying that this was the rate of reaction, right? And then we would go, so let's say this is the 4 degree, and then we started going, and then all of a sudden nothing, right? It just straight up stopped working. Okay, so this would be 4 degrees, and it would be warming up, right? Out of the refrigerator, it's going to start warming up slowly. And then in our reaction, we saw 40 degrees. Let's put 40 degrees right there. And then right here, when it hit somewhere, for us, the only data point we have is 90, it stopped working. Okay? So what that means is this enzyme likes to work somewhere close to there. Okay? Likes to work means it has an optimal, it has a preference, okay? It has an idea, okay, or it's structured to work at a certain temperature, okay? So optimal temperature for catalase, this enzyme, is somewhere, for our reaction, the only information we have would be somewhere close to here. And as this one got to about the same temperature after sitting here for a while, right, it's doing about the same. So optimal temperature is where that enzyme works most effectively or most efficiently or works the best. Okay? Okay? Optimal. Oh, this room is optimal temperature, right? It may not be for your friend who's like, oh, this feels like an Arctic tundra, right? This room feels good for you, right? This temperature is where it works best for this enzyme. Okay? So optimal temperature, make sure you know that term. It's not typed in your lab, okay? But optimal temperature, where that enzyme likes to work, okay? What it's designed for, okay? This one, catalase, is designed for about 40 degrees Celsius, okay? All right, the next one. pH. Erase all this. So the next one talks about, the next one is going to talk about how does pH affect the enzyme. Okay? And I kind of started talking about this a second ago when I was talking about enzymes in our stomach, in our gastric, okay, in our intestines, mostly in our stomach. Our stomach gastric enzymes, our pH work best about 1 or 2, okay? But if it's in our blood, that pH needs to be about 7, closer to neutral, okay? And so they're designed for specific ones. And so we're going to do an experiment to see where catalase likes to work, okay? And so we're going to set up our 3 test tubes, okay? 1, 2, 3. And we have, in the first one, we have the same reaction that we've been doing. We add a little bit of water, okay? So we have the enzyme and hydrogen peroxide. And so just like in the last one, okay, 8.1 and 8.2, we did catalase and hydrogen peroxide and we saw bubbles, okay? So we're gonna have that one. We're gonna add HCl, we're gonna add an acid, okay, hydrochloric acid, okay, make it acidic. And then we're going to add NaOH, okay, sodium hydroxide, a base, so we're gonna make it basic. So does it work here? Water is pH of close to 7, okay, neutral, okay? And then we have acid and then we have base. And so we're gonna fill that in. Water, and this is somewhere close to 7, okay, somewhere close to neutral. With an acid, okay, hydrochloric acid is usually 1 or 2, okay? Low numbers on the pH scale are acidic, so below 7 is acidic. The hydrochloric acid is very acidic, so 1 or 2. And then NaOH, okay, I'm gonna go ahead and put 14, okay? And that's the high number, but high numbers above 7 are basic, okay? And so we're gonna put those in, okay? And we would test that with pH paper, okay? We would tap it in and then we would test that with pH paper, but we're gonna go ahead and fill it in our video, okay? All right, and so now we're gonna do this reaction to see what we get. Does it work in all of these or not? Okay, so test tube 1 is our reaction that we've seen. Here is our test tube with the water and the catalase already in. And we're going to add hydrogen peroxide. And we should see bubbles. Hear bubbles. Okay? We see bubbles. That's what we've seen already. Okay? If I already added in the HCl, this is in acidic. Okay? No bubbles. And this is in basic. Also, no bubbles. Also no bubbles. So what does that mean? That means that catalase likes to work at a pH or close to a pH of 7. So in test tube 1, we saw a lot of bubbles, high amount of bubbles, high amount of oxygen gas being released in the breakdown of hydrogen peroxide. Okay, so that means catalase is working. Okay, is working means it's helping facilitate that reaction to bring hydrogen peroxide and then break it down into water and oxygen gas. That's why we're seeing bubbles. Okay. What does it mean in test tube 2 when we didn't see any bubbles? Okay, we didn't see any bubbles in this. If we put it in hydrochloric acid, we put it in an acidic environment. Okay, I'm gonna put this acidic environment. Okay, so we put that enzyme and that reaction to occur in an acidic environment, and we didn't see any bubbles. That means catalase is not working. Okay, so I'm trying to write each of these charts a little bit different so you can see that there's a lot of ways to write that data. Okay, so in this one, we're seeing it not work. Okay, why is catalase not working here? Well, the environment is acidic. Okay, catalase obviously by our data doesn't like acidic. Okay, because that acidic environment probably and did denature it. Okay, so it took that folded catalase enzyme and that environment broke those hydrogen bonds. Okay, and so it made it unfold, or maybe it folded differently because of that. Okay, so it changed the structure of that. This acidic environment is going to have a lot of hydrogens, and so it's going to break those bonds of that catalase and change it. Okay, so it's going to be denatured. Okay, it's not working because it's denatured. Okay, it's not in its natural state anymore. Okay, it's not in its normal folded lock and key form to bond with hydrogen peroxide. Okay. NaOH tube, test tube number 3, we saw no bubbles in test tube 3. This is NaOH is basic. Okay, this is a basic environment, sodium hydroxide. Okay, it's going to do basically the same thing that we saw for the acidic. Okay. It's also going to have denatured catalase, and we saw no bubbles. Catalase is again not working. Okay, catalase is again not working because of this basic environment. You're changing the OH concentration. Okay, and so that is also going to change the pH. So enzymes, like they prefer certain temperatures when we looked at the previous experiment here, okay, they also like a certain or a specific pH. And so you're going to have an optimal pH that they like as well. Okay? So optimal pH is the pH environment where that enzyme works most effectively or efficiently, or works best. Okay, I'll keep it short of enzyme. I'm almost done now. Okay, so optimal pH, okay, wherever that pH is, and based on our data, catalase likes it close to 7, close to neutral. It doesn't like it acidic, and it doesn't like it basic. Okay, so if we were to draw a graph, okay, it's going to work best at the optimal, okay, and then close to that on each side of it, it would work a little bit, but not a whole lot, okay, on the basic side, okay, and then the same thing would happen on the acidic side. I probably could have reversed those, but we get the idea, okay. So basic numbers are high, okay, and acidic numbers are low. That's why I was saying I probably could have reversed that. So let's put 1 to 2, because that's what we have on our chart, okay? And let's say this is 7, so this is our pH, okay? And this is enzyme activity, okay? Or reaction rate, okay? Okay, and so we're looking at this going, well, it likes to work right here the best. The top of that chart, that graph, is the optimal pH. That's where it works best. And then maybe there's a little bit of a buffer on each side where it can still work, but then dramatically will taper off until it does not work at all, and it does not work at all because it's been denatured on both ends. Okay, so that is our lab for today on enzymes. If you have any questions,